Today, we will be discussing with Dipshay Chand his recent PLOS ONE Lab Protocol article entitled Absolute Quantification of Neuromelanin in Formalin-Fixed Human Brains using Absorbance Spectrophotometry and the associated protocol in protocols.io. Dipshay, thank you so much for being with us.
Thank you for having me.
Dipshay Chand graduated with a Bachelor in Biomedical Sciences in 2023 from the University of Auckland in New Zealand, receiving first-class honors. Dipshay's honors research focus on developing methods to analyze neuromelanin and metal concentrations in the human brain in order to further the development of biomarkers to detect Parkinson's disease early. Dipshay intends to keep researching Parkinson's disease as a PhD student in the near future. When Dipshay is not working in the lab, he enjoys nature and portrait photography, playing chess, hiking, and playing video games. Dipshay, again, thank you so much for your willingness to dig a little bit more into this manuscript. To kick off with the conversation, can you briefly describe or explain what the protocol is about, how to use it, and why it is relevant?
Yeah, sure. So I guess I'll sort of start with the relevancy aspect of it. So as you know, in PD, in Parkinson's disease, sorry, we have the degeneration of the substantia nigra. And the substantia nigra is this really vividly black area in your brainstem. And in Parkinson's disease, we sort of see that vividness decrease. And that's just due to all the neurons there that are dying off slowly. And what we see is that these neurons are pigmented because of this pigment that we call neuromelanin. It's kind of like the melanin in our skin, but it's just found in the brain, so we just call that neuromelanin. And the thing about neuromelanin is that we don't really understand it that well. We don't really have an idea of how it's made. We don't really know why it's there. We have some ideas, but there's nothing really concrete. And especially we don't really know how it's affected by age, race, and sex. So we know that it might increase with age, but we're not really sure what happens at sort of the higher end of that. And in addition to that, as you mentioned earlier, that my honors project was actually based on looking at neuromelanin MRI, um, and how we can use it as a biomarker. Sorry. So with neuromelanin, some researchers in the past have found that you can actually detect it using MRI. And what this sort of leads us on to is potentially we might have a really easy biomarker. We can put someone in an MRI and potentially have an idea of maybe this person could get Parkinson's, maybe they could not. And that's just because the degeneration starts many, many years before we actually start seeing the really problematic symptoms, such as the shaking of the limbs and issues such as that. But the thing is, the research behind this isn't really clear in terms of what neuromelanin is doing. We know that the MRI signal changes, but we don't really know what neuromelanin is exactly doing in terms of concentration. So we've got this problem of we don't really know how it changes, like with age and in diseases. And then so the problem becomes we don't really understand it. So what do we do? So the first step is let's try and quantify it. Let's try and measure it so we can try and compare it between different ages, different sexes, different races, and it's most importantly in disease. But the issue here becomes that there's not really any methods to quantify neuromelanin. There's some microscopy methods, but those don't really focus on neuromelanin. That's just more looking at how light passes through the tissue, which is great, but it's not directly measuring our molecule of interest. So what our protocol does is it allows us to do exactly that. It's measuring neuromelanin, it's quantifying it. And just in terms of briefly how it works, it's quite straightforward. You would just slice up a bit of the substantia nigra, and you'd run it through a protocol. And I'm simplifying a bit here, but we basically heat up the piece of tissue in some SDS and some other chemicals, And then that basically just breaks down the fixation because the fixative essentially cross-links your proteins. So the heating up the tissue breaks down that fixation. And then the most important part here is then we digest using proteinase K, and that basically just breaks apart everything in the tissue, but that leaves behind this pellet of neuromelanin. And then with that, we just do some washes, purify it, and then dissolve it in some sodium hydroxide. And then we measure the absorbance, and that's how we get to the concentration. And that's basically all we need to do to quantify it. And yeah, so it's a relatively straightforward protocol. And the idea is that hopefully anyone can use it to get a quick measure of neuromelanin.
So I understand what the motivations for developing these protocols were, but can
Yeah,
you help us to understand the relevance? Can you also help us understand what was the state of the art at the moment? So, in other words, why previously published protocols to quantify neuromelanin didn't work for you? Why you have to create this specific protocol?
right. So actually, there weren't any protocols to quantify neuromelanin. There was, I believe, one, which is what we've sort of based ours on. But that was in fresh frozen tissue, and in fixed tissue, we've got all this cross-linking. So we tried, but it couldn't directly translate over to fixed tissue simply because the protease K, I believe, just couldn't digest it out properly. So for us, we needed a way to use a protocol for fixed tissue. And that's just simply because fixed tissue is a lot easier to store and keep around than fresh frozen tissue is. And there's just a lot more of it available, I believe. And yeah.
And digging into the protocol a little bit more, can you tell the users what are the most critical steps in your protocol?
Yeah, so the most critical step would probably be the proteinase K digestion. That is the step that actually sort of separates the neuromelanin from everything else. So if the timing or the temperature is incorrect, you're not going to get that efficiency of digestion, and in a way, you almost waste your precious tissue, because there's no sort of going back from it. And yeah, so it's just ensuring that your enzymes are up to standard and that you follow the timing and temperature requirements so that all the neuromelanin is digested out.
And while you were developing and optimizing the protocol, was there anything that you tried as a guess and didn't work and didn't make it to the final version of the protocol?
Yeah. So a lot of the optimization that we did, so basically taking the older protocol that was for fresh frozen tissue. So there was a lot of little changes, such as centrifugation speed, time, and temperature for the Proteus K incubation. But one of the things, yeah, so those are all things that we tried, but that was more just a higher temperature or a lower temperature or changing time. But we tried different washes with different non-polar solvents. So we actually ended up using acetone, but we tried things like methanol, ethanol, and chloroform. And these, for some reason, they just simply didn't work. Theoretically, they should have had, but for some reason, it just didn't. And that's sort of one of the mysteries around neuromelanin. And why it didn't work, we don't really have any idea, but acetone is the one that ended up working.
And it is my understanding, as we have discussed this, that you want to pursue your research focusing still on Parkinson's. So my guess is that you will be using this protocol, or you are currently using this protocol still. Have you had to troubleshoot it any further since the time of publication?
So I actually haven't actually used the protocol since publication. But the idea is to use it, yeah, for a much larger data set. For example, maybe 20 brains. But no, I haven't had to troubleshoot it just yet. But while I was, uh, when I did get the data for the for the data that was published in the article, there wasn't any issues that I came up with, and this simply might have been a matter of experience because I had done it so many times. So, and personally, no, I didn't have any troubleshooting issues, but I would be interested to see what others come up with.
And this might just be a guess. I know that you focus on human tissue, but do you get the same phenotypes when you mimic Parkinson's in animal models? And could this method be extended to animal models?
Yes, so actually, in animal models, so for example, mice don't actually have neuromelanin. So their substantia nigra is not dark like ours. Um, but there have been some recent papers, as in recent as this year, that have come out and they've sort of demonstrated a neuromelanin model of Parkinson's disease in mice. And so they do show that sort of age-dependent increase in neuromelanin. So protocols such as this could be really useful in potentially testing out like neuroprotective therapies in those mice to see how it affects the neuromelanin concentrations.
I see. And so, and now changing gears a little bit, you went to the whole process of optimizing the protocol, working to get it work, trying different solvents and so on. What was the motivation to first put it publicly in protocols.io, but then going a step further and have it peer reviewed in PLOS ONE and published as a standalone methodological paper?
Yeah, yeah. So when I was initially brought onto this project to look at this issue of measuring neuromelanin and measuring metals, there were a lot of methods to measure metals. That was quite widespread. But neuromelanin, that was very scarce. There was that one protocol that we had, but there was basically nothing for neuromelanin. So probably my biggest motivation was to get out a well-written method for formalin-fixed brains and yeah, just see what other researchers do with it because I guess the beauty of science is when we collab. Yeah, so to see what others with brain tissue can get from their brains using a protocol such as this.
And can you share with us how or what, how the feedback that you received from the peer reviewer shaped the final version of the protocol?
Yeah. The peer reviewers feedback was excellent. One of the yeah one of I think one of the most significant feedback that we received was basically to verify the basically elemental composition of the neuromelanin. So that's something quite relatively straightforward that I just missed during the process of this. But I guess yeah, seeing that feedback sort of just clicked like, ah, yes, that would be really useful in sort of verifying that yes, what we have made in a protocol was neuromelanin because we synthesized some neuromelanin for this protocol to act as basically a standard curve. So it was really helpful in just verifying that yes, what we have made is similar to human neuromelanin.
I see. And finally, I think that my last question would be, I know that this paper is relatively recent, but as a very early career researcher, can you tell us what do you think the impact is or will be of this manuscript on your uh, career?
Yeah, the impact of this was is and was huge. Yeah, this was my first ever published paper, and I was the first author on it. So just seeing that published was an excellent and just an amazing feeling. Yeah. And as I said, yeah, it is quite recent, but I think it was a really important first step in my research career. And yeah, I'm just hoping for more publications from here on.
And before we wrap up, is there anything that you would like to mention or discuss that we haven't touched so far?
Um, there's nothing I can think of. Oh, except I think we could maybe talk about I think one of the most difficult aspects of the protocol probably isn't mentioned, but one of the things that I struggled with somewhat in this was the pipetting steps can get a bit difficult. So to anyone trying to try this protocol, just be really careful with your pipetting because the last thing you want is to suck up your neuromelanin into the pipette and that's your sample ruined. So yeah, just be really careful with that and just be wary of how much suction pressure you're applying. Yeah,
So in addition to the digestion of the tissue
yeah.
using proteinase K, the pipetting, it's a critical and step
Yes.
that you need to clearly control.
Yeah. And it's just the fact that there's so much pipetting because there's so many different washes and you do have to be really careful and really consistent with it. We try things like not pipetting to avoid sucking the pellet of neuromelanin out, but the issue becomes if you try and pour it out, you get a little bit of the liquid just stuck to the bottom just because of the capillary action. Yeah.
Okay. Thank you so much, Dipshay. This has been like a enlightening conversation. I have enjoyed it, and I know that it's going to be useful for the users of the protocol.
Thank you so much for your time. Yeah. Thank you for having me.